gonna talk a little science. I'm gonna teach you guys some science. I know some people, who's hyped about science? Yeah! That was like the fakest cheer I've ever heard in my life. Now, why? No. I know you guys like science. We nerds in here. That's why. That's how we get down. We all nerdy. We all mad nerdy, so it's cool. So I'm with family. I'm with community on the nerdy part. So I'm gonna explain to you guys just in the next like 8 minutes. I'm trying to explain to you one piece of technology that I've been working on. Okay, trying to teach you one thing. Hopefully you will learn a few kind of interesting concepts from this and like hopefully understand what I'm saying. Okay. And I should caveat this with I don't know what's gonna happen next when I press these buttons. So hopefully it's something cool. All right. So any of you have any idea what that is? Any idea? No idea. Just no. Eric, do you have any idea what that is?
I can't see it.
You can't even see it. That's how this.
Oh,
this?
That's.
Yeah, you know what that is, right?
Yeah.
Yeah, you got that. Don't tell anybody though. Just keep it to yourself. Just keep it to yourself, because you don't want to impress everybody with your knowledge. You know what I'm saying? So, and if you don't know what that is, that's cool, because I actually didn't know what was until about 2 months ago, and I still don't quite know what that is when I look at it. But this is gonna be like The Usual Suspects. Okay, I'm gonna show you the end of the movie at the beginning, and then we're gonna go through the whole movie, and at the end we're gonna go back to the beginning, and you're gonna be like, oh shit, now I know what it is. Okay, that's my goal. Okay, so we're gonna make this like Usual Suspects. Okay, and I don't know who Kaiser says it is, but anyways, all analogies fall apart at some point. Okay, so the first thing I'll say is this thing is a diagram. Okay, all of those little symbols and parts, they represent DNA. You guys know what DNA is, right? We got a basic language on there. Everybody knows what DNA is. Okay, so this thing that I've circled, MIRX. MIRX stands for something called a microRNA, okay? A microRNA. I don't think anybody here has heard about what that thing is, okay? So microRNAs, they're... Oh, we got a hand in the back there. All right, thank you. Thank you. You got at least one microRNA nerd. More, good. Ah, Chuck Vine knows a microRNA. Really? You know, I didn't even know what microRNAs were like 2 months ago. Crazy. Okay, cool. Smart people. All right. So basically, this is a microRNA. MicroRNAs are really interesting for lots of reasons. So DNA is kind of like the hype man, or sort of like the main act of a performance. It's like the Will Smith, and always got the spotlight. Everybody's talking about DNA. MicroRNAs are kind of like, the unsung hero. They're kind of like the DJ Jazzy Jeff of biomolecules, okay? You can't have Will Smith without DJ Jazzy Jeff, and you can't have DNA without microRNAs, okay? So basically, just for the rest of this, that's what you gotta think about. Okay? Boom. Alright? That's DJ Jazzy Jeff. He's the microRNA. Okay? I gotta say it too. I spent so much time figuring out which animation to choose. I was like, there's like one where there's like a comet that like flies by and then Jazzy Jeff appears. I was like, do I want the comet or like the exploding lens flare? Anyways, whatever. These are the things that I think about. Okay, so that's that. Okay? So now you know what a microRNA is. Now, microRNAs are really crazy. They're really, really crazy, and they're amazing. So the thing that's neat about microRNAs is there are about 1,000 of them in our bodies. Like every single cell in your body right now, you've got about 1,000 different microRNAs that are floating around. And what's really, really interesting about them is if you only have a few of them, just like 5 or 6 of them, just a handful, okay, you can actually, if you know the amounts of the microRNAs that are inside your cells, okay, just a specific 5 or 6, you can create a little fingerprint, okay? In other words, I can say, if I just know these 5 microRNAs, the amount that's in them, I can say this cell is a cancer cell versus this cell is a healthy cell, okay? And even more, and this is something we're just starting to learn more about this, so this is very much kind of research happening right now in the lab. We're just starting to figure out not just differentiating, say, between healthy cells and cancer cells, but say different types of cancer cells, or different levels of cancer, so how sick you are, okay? So these molecules are extremely important indicators that we can use as fingerprints to try and identify and classify different types of disease states, basically, okay? Just these little molecules, okay? They're crazy, they're really interesting for lots of reasons, but for today, just think about them as a fingerprint, all right? Now back to this thing, okay? So what the hell is this crazy thing? And what this is something called a genetic circuit. Okay, genetic circuit, 2 words, 1 genetic, 2 second circuit. This thing is made out of DNA, okay? It's a DNA circuit, and when I say circuit, what I mean is, it's a computer, okay? This DNA is a little computer, okay? And I think a little, yes, that's a computer, okay? So what happens is, you take this little computer, and you put it inside a cell, you can ask the cell questions with this piece of DNA, okay? And the question with this particular circuit you ask is, hey cell, it's like, hey girl, how much microRNA do you have? Genetic circuits don't talk that sexy though, so just keep that in mind also. But that's what they can do. You can put these things into cells and ask the cell a question, okay? And this particular circuit is asking how much microRNA do you have, all right? And then, so this thing is all crazy. Like, what is that? That's even crazier version of a different type of circuit. And if you're really kind of clever, you can design these circuits to ask not just how much of one microRNA, you can ask, say, 5 different microRNAs if you build a complex enough computer. And like I said before, if you know only 5 or 6, you have a blueprint. You have a fingerprint where you can identify a cell, right? So if you can develop a complex enough genetic circuit that can ask multiple microRNAs, you can ask questions like, hey cell, are you cancer? Okay? And so that is the technology, one of the major technologies that I'm working on, is how to build these circuits to ask cells questions, okay? So this was a collaborator on some friends, I was gonna say some homies, they are homies, but you know, they're collaborators in science speak. It's so funny, man, I like, you know what I mean, I'm like in lab all day doing my thing, and then I come here and I'm like, yeah, you know, my homies in lab, I'm like, no, they're not homies. So what these guys, my collaborators did recently, was to create a little DNA computer that they could inject into a cancer cell and ask that cell, are you cancer? And if it was a cancer cell, on top of that, it would initiate a process called apoptosis, which basically means cell death. It would actually kill the cell. So you can put that little computer into a whole bunch of different cells, and it'll know, just by asking the cell that question, if it's cancer or not. And if it is, then it'll kill that cell, okay? So this is one type of cancer therapy that's being developed. Now, this is one interesting application. But for me, what I'm working on now, what I'm interested in, is just not one of these circuits, but I want to build libraries of these circuits. Lots and lots and lots of circuits. And the reason why we're interested in building libraries of these circuits is because we don't want to just ask these cells, are you cancer? That's a really good, important question, but we want to ask them all kinds of interesting questions, like, how rapidly are you dividing? Like, why are you so depressed right now? You know, whatever. Whatever kind of question you want to ask, we want to be able to ask that cell. And you need huge libraries of these circuits to be able to ask the cells the types of questions we want to ask them, okay? So what I'm working on now is building these libraries, okay? So, you guys with me? You guys with me? All right, cool, cool. So, you guys can re-explain this to me later. Hopefully this makes sense. So, closing it out, the next question is, how do we build these circuits? Okay, so I've explained the biology, what we do with these little DNA computers. The next part is how we build these circuits. And so, this is a whole, I wish this wasn't so washed out, but anyways, this is a crazy looking design. And it's funny for me actually too, because as I've kind of gotten into science in this field that I'm in, there's actually a lot of really visual beauty to a lot of the kind of engineering and devices that we make, and for me, I didn't even think about this until now, but I really enjoy, these things are called microfluidic devices, I'll explain more in a second. But you can inject these things with food color, and that's like my favorite thing to do, is fill them up with food color and like look at them with a microscope. I didn't even think about this until now, but I like taking pictures of these things basically. So, weird, okay. And so basically, this thing, this little device, you can kind of see my fingertips, it's about the size of a postage stamp, okay? So all of those little tiny squiggly lines are like, you know, microns, 10 to the minus 6 meters, tiny, tiny, tiny little lines, fraction of a, you know, width of hair. And basically, you can think about a microfluidic device as a miniature factory, okay? So, you know, back in the 60s, we had computers that were massive. They were the size of the room. They couldn't tell you that 2 plus 2 equals 4. We miniaturized computers, and now they're everywhere. You got them, everybody's carrying a computer with them, okay, everybody. So, microfluidic devices are the same type of thing, except miniaturizing biology. We're taking big biological systems and miniaturizing them into little tiny chips. And so these things actually are made of this material that's the same stuff that you find in silly putty, actually. And what's crazy about these chips is that you can basically execute all these crazy operations. So by having multiple layers of patterned, essentially the silicone rubber, you can create these very complex devices that can manipulate fluids and essentially have these very, very precise factories that are, again, you can run hundreds, thousands, tens of thousands of parallel reactions in these little tiny chips that are the size of a postage stamp. And what we're doing is to basically take these devices, so these are kind of devices that I built that can synthesize these different types of, they're called fluorescent proteins, they're glowing proteins. So we can make proteins with these devices, and now, okay, so full circle, I think you guys know what the punchline is going to be, right? Kind of, I've been leading you guys into it. We're going to use crazy devices like this, so this is one that myself and one of my homies created the other week, to basically build these circuit libraries. Okay. claims DJ Jazzy Jeff. Okay. So that's it. That's it. So basically, that's what's up. This is one of the projects that I'm working on that I personally am really excited about. Is creating these DNA library, these DNA circuits, these little DNA computers, and building them with these chips. Okay.
Yeah.
That's it.